Greetings, this is Team Amboborella Trichopoda's presentation on the study in regards to the polyphyly of Asian tree toads of the genus Pedostibes Gunther 1876, and the description of a new genus from Southeast Asia. The focus of the study is on the Asian tree toads of the genus Pedostibes, which consists of the following allopatric species: Pedostibes hosi, Pedostibes tuberculosus, Pedostibes kempis, Pedostibes rugosus, and Pedostibes everetti. So, for some added background on some of these species, let's start with the Pedostibes tuberculosus. Pedostibes tuberculosus is a member which is restricted to the western Ghats of India. The female members of this species exhibit a maximum size of 41.5 millimeters, with males having a maximum size of 38.2 millimeters, as recorded in this study. The next member of this genus is the species Pedostibes kempi, which is only found at the Garo Hills on the northern eastern region of India. Originally reported to have a body size of 34.0 millimeters, has not been encountered after the initial description. So the next toad involved in this study is the yellow-spotted climbing toad, or the Pedostibes hosi, which is only situated in the Thai Malay Peninsula. Females have a maximum body size of 104.8 millimeters, with males having 79.6 millimeters as their maximum body size. Now this is the Pedostibes rugosus, which is a species restricted to the island of Borneo. Now, due to the presence of 8 presacral vertebrae and the possession of cocci, these two, this and P. everetti, were reassigned from the genus Nectophyrne to the genus Pedostibes. Now, the females have a maximum body size of 95.0 millimeters, while the males of this species have a maximum body size of 76.8 millimeters. Now, like the previous species, the Pedostibes everetti is also restricted to the island of Borneo. Its females have a maximum body size of no more than 95.0 millimeters, while its males have a maximum body size of 76.8 millimeters. However, this species has no recorded adult specimen yet.
This study was performed due to the following reasons. First, genus Pedostibes exhibits disjunct distribution or allopatry, which causes questioning regarding its biogeography and taxonomy of the group. Second, the need to re-examine taxonomic status of both Indian and Southeast Asian Pedostibes to determine their polyphyletic relationships and to see if it can be corroborated with other sources of evidence. This evidence being the morphology between the species and their modes of reproduction. And last is the need to define a new genus that reflects taxonomic placement and unique evolutionary history for the Southeast Asian species. Why for the Southeast Asian species? Because P. tuberculosus has priority as the type species. Figure 1 shows the map distribution of the species of Pedostibes endrantapia gen. It can be seen in the figure that the species are concentrated in India and some parts of the Southeast Asia.
Materials and methods. This part of the study is divided into 3 sub-parts: morphological analysis, phylogenetic analysis, and nomenclatural acts. The first one is morphological analysis. The figure shows the list of specimens examined. Due to the unavailability of specimens for P. campi and P. everetti, only the male species of P. tuberculosus, P. hosii, and P. rugosus were examined. With the available specimens, the value of the following was obtained using mid-tutorial digital caliper: snout fin length SVL, snout length SNL, head length HL, head width HW, tympanum diameter TD, forearm length, FAL, third finger length, fin 3L, third finger disc width, fin 3DW, tibia length, TBL, and inner metatarsal typical length, MTTL. The measurements mentioned were corrected for proportional variance by individually dividing each character by the specimen's snout vent length, or SVL. This is due to the large differences in body sizes. With the data gathered, 3 types of analysis were performed. A. Analysis using statistical software environment, R.V.3.1.2. B. Principal component analysis, or PCA. And C. Discriminant analysis of principal component, or DAPC. Analysis using statistical software environment, R.V.3.1.2. This analysis was conducted for adjusted measurements by which simple mensural comparisons were performed by plotting each character against the adjusted measurement. The second is principal component analysis, or PCA. This analysis was performed to find the best dimensional representation of morphological variation and whether this could provide a statistically sound and detectable group structure. Principal component with eigenvalues of 1.0 or more were retained based on the Kaiser's criterion. The last is the discriminant analysis of principal components, or DAPC. This analysis was performed using all-package adjuvant 2.0.0. This was also performed to characterize clustering and distance in morphospace by locating possible linear combinations of morphological variables that possess the largest between group variance and smallest within group variance. The second part of the method is phylogenetic analysis. The table shows the sequences of 1 mitochondria, 16S, and 2 nuclear markers, CXCR4 and NCX1, that were aligned using the muscle algorithm. 2 of the major analyses performed in this part are maximum likelihood phylogenetic analysis and Bayesian analysis. Maximum likelihood phylogenetic analysis. This phylogenetic analysis was performed with the program RAXML using GTR plus gamma nucleotide substitution model, in which node support was assessed with 500 bootstrap replicates using rapid hill climbing algorithm. Bayesian analysis. This analysis was implemented in the program MrBayes 3.2.6 using 2 independent runs, 4 chains each, with MZ chains in the length of 50 million generations per run. The last part of the method is nomenclatural acts. According to this, new names in this article conforms to the International Code of Zoological Nomenclature. The nomenclatural acts herein are registered in ZooBank, the online registration for ICZN. Lastly, the electronic edition was published in a journal with an ISSN, which has been archived and available from digital repositories.
The results of the study are distributed into 5 different categories. The first one being analysis of morphological variation, second phylogenetic analysis, third natural history, fourth systematics, and the fifth description of new genus. Based on the morphological analysis, it was shown that there is a clear and complete separation between P. tuberculosus and P. horsei plus P. rugosus for every morphological variable as shown on Figure 2. We can clearly see on these scatter plots the clustering of the circle and triangles, while the squares are separated from them. P. horsei is represented by the circles, P. rugosus by the triangles, and P. tuberculosus by the squares, meaning that P. rugosus and P. horsei have similar characters while P. tuberculosus is different. The characters HW, FAL, FI, and FIN3L explain the most variation along the PC1 axis, and the characters FIN3DW and FL for PC2 showed heavy loadings. The results for the DAPC analysis showed exclusive clustering between all three species. For the phylogenetic analysis, both ML and Bayesian phylogenists recovered similar topologies, which means they have the same branching pattern, with the ML tree having higher resolution at deeper nodes. As we can see here, P. tuberculosus formed a sister clade relationship with the genera Adenomus, Xanthophrene, and Dutaphrenus. Males from this clade tend to be small, except for the genus Dutaphrenus. We can also infer from the bootstrap values that the support is high. Southeast Asian Pedostibus here is shown to be reciprocally monophyletic with the genus Phrenoides, meaning that the haplotype from the genus Pedostibus are more closely related to each other compared to the other genus Phrenoides. The bootstrap value shows a 0.6 to 93 support for this proposed new genus. Additionally, this clade represents the largest body size among South and Southeast Asian toads. Both South and Southeast Asian Pedostibus are forest stream associated taxa, but P. horsei is primarily a lowland frog, while P. tuberculosus is a mid to high altitude species. The breeding of P. horsei and P. tuberculosus is similar. The male frog clasps the female frog from behind the forelimbs. The oviposition mode is different, however, in that the eggs of P. tuberculosus are laid as films, while for the P. horsei, the eggs are laid as long strings. The reproductive behavior of P. kempi, P. rugosus, and P. everetti are unknown because of the lack of specimens. The current taxonomy that unites the South and Southeast Asian tree toads is not supported by the following evidences gathered. First, distribution. Second, morphological. Third, natural history. Fourth, phylogenetic relationships. And fifth, genetic divergences. So what they did is that they assigned the Southeast Asian lineages to a separate genus, Rentapia. The Indian species P. tuberculosus will retain its genus since it has priority as type species. The taxonomic status, however, of P. kempi and P. everetti remains unresolved due to the lack of samples. Additional data is needed to resolve their taxonomic placement. P. kempi is placed temporarily under the genus Pedostibus, while P. everetti is assigned to the new genus Rentapia. The reason being that P. everetti is phenotypically similar to P. rugosus, with the only difference being the presence of a tarsal ridge. Should additional data become available, the relationship of the toads will be re-evaluated again. The description of the new genus Rentapia is summarized into 4 different parts. Diagnosis: large-bodied toads with a movable coccyx, 8 pre-sacral vertebrae, a complete quadratojugal, and small pigmented ova laid as strings. Adults are primarily arboreal and inhabit riparian vegetation around small to moderately sized forest streams. For the phylogenetic definition, Rentapia is a node-based name that refers to the clade originating from the most recent common ancestor of Pedostibus horsei and Pedostibus rugosus. For the content, P. horsei and P. rugosus renamed to R. horsei and R. rugosus. P. everetti tentatively placed under Rentapia due to its close geographic proximity with the other 2 species. And lastly, for the etymology, epithet is selected to honor the legendary warrior and Malaysian national hero Libau Rentap of the Iban, an indigenous group in Borneo. To summarize the discussion, Pedostibus is small in size and deposits medium-sized clutches of eggs that are laid as films. The combination of these traits differentiates it from the Rentapia genus, which is diagnosed by its larger body size, and an oviposition mode characterized by a large clutch of eggs laid as strings. Male and female Pedostibus have the same color patterns. Our Hossiae females are vividly colored. There are no evidences yet of color dimorphism for R. rugosus and R. everetti due to the lack of specimens. And lastly, Rentapia and Phrenoides have a striking morphological and ecological difference, so it is best to consider these two as a separate genera, even though merging these two would solve the polyphyly of P. sensulato. These are the sites that we used in order to gather the images of the specimens. And that ends our presentation. Thank you for listening and stay safe.